Okay, hi psychology friends. We're going to talk today about Unit 3 A, B, and C because we've got a test coming up. So let's take a look at what we've covered and review. As you know, this is the biological basis for our behavior, and we'll be talking a lot about the brain and its basic building blocks. Neurons are the building blocks of the nervous system. There are three types of neurons, as you know. Motor neurons, which carry impulses to our voluntary and involuntary muscles throughout our body. Sensory neurons, which carry information from any of our sensory points on our body to the brain. The point at which they meet are called interneurons, and these interneurons are located exclusively in two places. That is in the brain and the spinal cord. Ooh, look at that fancy spin. All right. All right, now we're going to talk about the anatomy of what makes these building blocks, the neurons. The longest part of the neuron is actually the axon. And this picture that we're looking at is a little flipped from the one that will be on the test and the one that we discussed in class, but is still a neuron nonetheless. Let's start on the left where we see the axon terminal. That is going to be the point that releases our neurotransmitters into the synaptic gap after it receives the electrical signal from the axon. Next, we have the longest part of the neuron itself, and that is the axon. The axon contains Schwann cells that help convey that electrical signal down the length of it. Some are very short, like the ones that go from our eyes to our occipital lobe. Others are longer that go all the way to the tips of our toes. Each of these little jelly bean shaped molecules is a myelin sheath. Myelin is the insulating material of the neuron. It helps make sure that that electrical signal that comes from the dendrite to the axon makes it all the way down effectively and without any issues. It's essentially like the insulation on the outside of a cord. It protects the degradation of that electrical signal as it travels down the axon. Two diseases that are affected by a breakdown in myelin are MS and Lou Gehrig's disease, which is also known as ALS. Both of these, as that myelin breaks down, that electrical signal conduction is inhibited. Over here on the right, we have the actual cell body. The cell body, or soma, is where the dendrite branches come off of. And at the center of that soma or cell body is the nucleus. That's where the DNA of the neuron is stored. The conduction of a current down an axon is known as, wait for it, action potential. And the minimum level of stimulation needed to activate an action potential is known as the threshold. Now let's look at a synapse so that we can know what's going on there. Synapses, as you know, are the connection point of dendrites and axon terminals. This is the chemical process by which neurotransmitters are released into that synaptic gap in between the two nerve cells. A on here is the mitochondrion, that's the DNA of the nerve cell. B is the vesicule. Vesicles store the neurotransmitters. C is the synaptic gap, the area between the two neurons. Psychotropic drugs and any medications actually work to inhibit the reuptake of those neurotransmitters back into the presynaptic neuron by keeping the neurotransmitters out in that synaptic gap either longer or for a longer period of time or for a shorter period of time. D is the receptor sites. It's good to know that these receptor sites are specific to specific neurotransmitters. Agonist and antagonist cells. Agonist cells molecules excite. Antagonist molecules inhibit. They are ones that look just enough like the neurotransmitters to fit into the receptor sites there, D. E are the neurotransmitters. In your book, there are a whole list of about 6 of them. Be good to know. Notice we're skipping F. X is the presynaptic neuron that sends the message, and Y is the postsynaptic neuron that receives the message. Once that chemical message is received, the postsynaptic neuron receptor sites release the neurotransmitter back into the synaptic gap or cleft, and it is taken back up into the vesicles through a process known as reuptake. Reuptake. Alright, here's just 3 of the neurotransmitters, and it's important to be able to match the function of those neurotransmitters to what they do. Let's look at the first one, GABA. GABA is a major inhibitory neurotransmitter. Undersupply is linked to seizures and tremors. ACH, we talked about this being used in surgery when you have anesthesia. ACH enables muscle action, so to inhibit that ACH conduction in the synaptic gap or cleft would cause muscles to not be able to react. So ACH is blocked and muscles do not move during surgery. Serotonin. Serotonin is used in antidepressants. It affects mood, hunger, sleep, and arousal. Glutamate. Glutamate is a major excitatory neurotransmitter. It's involved in memory. You've probably heard of glutamate. It's the G in MSG, a food seasoning located in many Asian foods. MSG here in America, it had a little bit of a backlash because they found a connection between MSG and migraines. The reason for that is it is an excitatory neurotransmitter. It causes too much brain action going on. These neurons are the building blocks, and they go on to make up the nervous system. The nervous system, as you see, is divided into 2 parts, the central nervous system and the peripheral nervous system. The central nervous system is the brain and spinal cord. The brain receives and processes sensory information, initiates responses to that, stores memories, generates thoughts and emotions, and the spinal cord conducts signals to and from the brain and controls reflex activities. Our peripheral nervous system is broken down into 2 types. Our motor neurons and our sensory neurons go right to the peripheral nervous system and control action. Our somatic nervous system controls voluntary movements, where our autonomic nervous system controls those that are involuntary, such as breathing, heartbeat, any of the things that we don't have to think about on a active basis. And if you want to think about it, autonomic, automatic sounds similar. But the peripheral nervous system has a somatic and an autonomic nervous system. The autonomic is further broken down into the sympathetic and parasympathetic divisions. Sympathetic are the things that activate and encourage and excite. Parasympathetic are the ones that calm it down. And in the book there's a good diagram of all of those different functions within our body. Language is a complex process, as we discussed, that is affected by many different areas of the brain. Each one, if they're affected, can cause a lot of problems. It starts out back on the back of that visual cortex. The visual cortex is located at the back on the occipital lobe. On this graph it is in red, so you see it there on the back. This information is then sent to the angular gyrus just above it. This transforms visual representations into an auditory code. The information is then sent down to the auditory cortex in the Wernicke's area. The Wernicke's area interprets that auditory code, turns it into a usable message, sends it onto the Broca's area, located just to the front of there, blue on this graph. The Broca's area controls speech, muscles via the motor cortex, sends that signals on to the motor cortex in an intelligent way so that they can pronounce and send signals to our muscles so they can know which vowels, consonants, sounds to utter, the degree to which sound will travel across our vocal cords. We talked about having 2 brains, our left and our right hemisphere. The dense band of neural fibers that connects the left and right hemispheres, as everyone should know at this point, is the corpus callosum. So why get a hemispherectomy? Why do some people have that split? Why not? It's covered by Obamacare. No, seriously, why would someone do that? Extreme cases of epilepsy or seizure. Epilepsy and seizure are essentially an electrical storm between the 2 hemispheres, and the information causes problems that we don't need. When you sever that thick band of neural fibers between the left and the right hemisphere, it stops communication between the two. It also causes some challenges for those that have hemispherectomies. Our left brain and right brains are very different in the way they process. Left brain focuses on language, analytical thinking, concrete, step-by-step functioning, where the right, as the woman who had the stroke in Stroke of Insight on the TED Talk, is about peace and harmony and our connectedness to the world and other, nirvana, right? Let's look at a few parts of the brain. This one controls emotional components that are sent through our sensory inputs to our brain. It's not a conscious process, but it is controlled by the amygdala. There's one on the left and the right. We're least conscious of this area of the brain down at the bottom that controls our involuntary muscles, heart rate, breathing, etc. Brainstem. The brainstem is known as the reptilian brain because it is the very most basic thing that needs to be there to keep us alive. It's important to know some differences between chemical messengers within our body. Two different systems that we have going on are our nervous system. The chemical messengers of the nervous system are neurotransmitters, and they're released into the synaptic gap. They're receptor proteins that reach their targets on the receiving dendrite. The other slower process is our endocrine glands. They release hormones. Hormones are the chemical messengers of the endocrine system. Some of these would be adrenaline. Adrenaline is released from our adrenal glands and gives us extra energy in times of fight or flight. Okay, this is a good overview of information that is going to be on the test. The only thing I'll point out on this slide is that hormones are carried by the blood system, our bloodstream, and that means that they're in the body longer. Neurotransmitters are released by the axon, so it's a much more immediate reaction, but it's also over quicker. Hormones are carried by the bloodstream and stay in our system a little bit longer. The only two other things I would ask you to be aware of for the test is what is evolutionary psychology. Try and think of a modern example of some trait that we have and explain how an evolutionary psychologist would explain away that based on the beliefs of evolutionary psychology. And the final thing I'll leave you with is when we talked about the genetic components of what makes up us, know what heritability is. Heritability, again, is the differences, the amount of weight given to our genetic factors rather than our environment. And we talked about nature versus nurture. If something has a high level of heritability, that means it is more directly controlled by genes in the population than in the environment. Another thing that would be good to study and know the difference between, we went over this a good deal in class, is the difference in twins, identical twins being genetically exactly the same, coming from one sperm and egg that was fertilized. They have the exact same genetic material. Fraternal twins being no more alike than any brother or sister. Psychologists find twins very interesting because they can study the effects of nature versus nurture in a more direct way. Thanks. Hope you do well on this test next week, and hope this review was helpful for you.